Hi everybody, welcome to your own YouTube channel vEgyan. This is part 2 of our discussion on Delhi University Biochemistry entrance exam paper of year 2020. We have provided explanation for each and every question. If you have any further doubts, you can drop them in the comment section or email us at vegyankur@gmail.com. Also, please do not forget to like, share and subscribe our channel to never miss an update. In the first question, the third strand of triple helix is paired in which scheme? The right answer to this question would be option B, Hoogsteen base pair scheme. A triple-stranded DNA, also known as the H-DNA or triple X DNA, is a DNA structure in which three oligonucleotides wind around each other and form a triple helix. So how this triple helix is formed? In the triple-stranded DNA, the third strand binds to the B form of DNA by forming Hoogsteen base pairs or reverse Hoogsteen hydrogen bonds. The answer is option B. The next question is the first three bases of 6-base recognition cleavage site of HindIII are AAG. What is the complete sequence of this 6-base pair site? The point that we have to remember here is that the restriction site of HindIII is a 6-base pair palindromic sequence that is read the same from both forward as well as the reverse direction. So the option B is going to be the correct answer as it reads as AAG CTT from both the sides. So option B is the correct answer. In the next question, what leads to the activation of protein kinase C? The PKC or protein kinase C consists of a family of closely related enzymes that play important role in several signal transduction cascades. PKC enzymes are activated by signals such as increase in concentration of diacylglycerol, which is DAG, or calcium ions. So the right answer to this question would be option number A, the release of intracellular calcium and diacylglycerol. The next question is asking about the transbilayer diffusion is also called as. So the transbilayer diffusion is also known as the flip-flop. When the phospholipid migration occurs from inner side of the bilayer to the outer side, it is denoted as flop. And when it is denoted as flip, when it occurs in the opposite direction. So the right answer is flip-flop, which is option number C. In the next question, the first step in the payoff phase of glycolysis is. So the breakdown of glucose, which is a 6-carbon atom molecule, into 2 molecules of pyruvate, which is 3-carbon, occurs in 10 steps of glycolysis, which can be further divided into 2 phases. The first is the preparatory phase, which comprises of step number 1 to 5, and the payoff comprises of step number 6 to 10. Here, the first step of payoff phase is oxidation of glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. So the correct answer for this option would be option number B. The next question is, what region of antibody binds to protein A during affinity purification? So it has been proved via crystallographic refinement that the primary binding site for protein A on the Fc region of heavy chain between the CH2 and CH3 domain. So the right answer for this question would be heavy chain within the Fc region. So A is the correct answer. In the next question, if oxidative phosphorylation was coupled in the mitochondria, then there is. So the uncouplers of oxidative phosphorylation in mitochondria, they inhibit the coupling between electron transport and the phosphorylation reaction. This will thus inhibit ATP synthesis without even affecting the respiratory chain and the ATP synthase. Hence, the final outcome would be increased oxygen consumption but halted ATP, and hence increased inorganic phosphate and ATP concentration. So the right answer would be option number C, increase in organic phosphate in the mitochondria. The next question is asking about the enzyme which is responsible for removal of supercoiling in the replicating DNA ahead of the replication fork is. So topoisomerases are the enzymes that are responsible for introduction and elimination of the supercoil. So topoisomerase is the correct answer for this particular question. The next question is asking that which of the following are not a DNA virus? Among the given option number B, which is the influenza viruses, are the negative sense single-stranded RNA viruses which cause influenza in birds as well as certain mammals. So the right answer to this question would be option number B, influenza viruses. The next question is asking the formation of one molecule of glucose from pyruvate requires. In gluconeogenesis, the pyruvate is converted into glucose, and the synthesis of one molecule of glucose from two molecules of pyruvate requires 4 ATP, 2 GTP molecules, as well as the NADH molecule. So here, the net equation of glyconeogenesis is 2 pyruvate plus 2 NADH plus 4 ATP plus 2 GTP, 6 H2O plus 2 H positive to give a glucose. So the right answer to this particular question would be option number A, 4 ATP, 2 GTP, and 2 NADH. In the next question, cyanogen bromide is used for cleavage of protein. The target site for cleavage is. So to answer this question, different reagents are used for the specific and efficient chemical cleavage of proteins in a solution, which cleaves at a specific loci. For example, cyanogen bromide, which is CNBr, it cleaves at methionine. Then there are several other, for example, formic acid cleaves at aspartic acid and proline peptide bond. Hydroxylamine cleaves at ASN-GLY peptide bond. So the right option for this question would be option number B, C-terminal end of the methionine residue. The next question is asking that who won the Nobel Prize in Medicine in 2018 for their discovery of cancer therapy by inhibition of negative immune regulation. So the correct answer for this particular question is James P. Allison and Tasuku Honjo. Both of them won the Nobel Prize in Physiology or Medicine for their approach of treating cancer by treating the immune system rather than the tumor. So the correct answer would be option number A. The next question is the biological role of restriction enzyme in bacteria is 2. A bacterium uses the restriction enzymes to defend against the bacterial viruses, also known as the bacteriophages or phages. When a phage infects a bacterium, it inserts its DNA into the bacterial cell, so it might be replicated. The restriction enzymes that are present inside of these bacteria, they prevent the replication of phage DNA by cutting it into many small pieces. The correct answer for this question would be option number C, to cleave the foreign DNA. The next question is asking that which of the following DNA sequence contain a 4 to 8 base pair palindromic site. Since palindrome is a sequence which is read same from the both direction, we have to check the complementary sequence of the given options. So among these options, option number B, the sequence reads the same in both directions. So the correct answer to this question would be option number B. In the next question, which of the following component is not a constituent of a typical A-tailing reaction? The A-tailing is an enzymatic method for adding a non-templated nucleotide to the 3' end of a blunt DNA. So we are going to need a blunted DNA. Now, generally, tailing is done to prepare the T vector for TA cloning. For this, the Taq DNA polymerase and clino-exo polymerases are used for DNA end modification. So the correct answer to this question would be B, ATP, which is not a constituent of typical A-tailing reaction. The next question would be the presence of salt during gel filtration helps too. The gel filtration chromatography separates proteins solely on the basis of their molecular size. The purpose of adding salt to the elutant is to minimize the secondary interaction or sample component with the resin. So the presence or absence of sodium chloride in aqueous elutant, it influences the elution volume of the protein by altering their hydrodynamic radius as a function of salt strength. The correct option is option number C, to reduce the non-specific interaction of the protein with the gel matrix so that the elution can happen effectively. That is why the salt is added to the gel filtration chromatograph. The next question is, HAT medium is used for hybridoma production contains the HAT media, or also known as the hypoxanthine aminopyridine thymidine medium, is a selection medium for mammalian cell culture, which is used in the hybridoma technology. And now, as the name suggests, it contains thymidine, which is option number C. So the option number C is the correct option. In the next question, which of the following classes of antibodies are expected to be immunoprecipitated predominantly using anti-J chain antibodies? So J chain is a protein component of both IgA and IgM. Hence, the presence of anti-J chain antibodies, IgM and IgA, are expected to be immunoprecipitated. Looking at the options given here, the correct answer would be IgM, which comprises of this particular J chain. Option B is correct. The next question is, which of the following antibodies is most efficient in causing agglutination? Again, IgM is the first antibody that is built during an immune response. So it is responsible for agglutination and cytolytic reactions due to its pentavalent nature, which gives it 10 free antigen binding sites, as well as it possesses a high avidity. The correct answer to this particular question would be option number A, IgM. In the next question, an unknown bacteriophage has a base composition of 23% of A, 36% of T, 21% of G, and 20% of C. Its genome is likely to be. Now, if you remember the Chargaff's law, in a double-stranded DNA, each of the DNA strand observes following approximate that percentage of A is almost equal to percentage of T, and percentage of C is almost equal to percentage of G. So in this particular question, where A is 23, T is 36, G is 21, and C is 20%, they are not aligned with the Chargaff's rule of double-stranded DNA. Hence, the DNA must be single-stranded, so the option number B is correct. In the next question, during the growth of mammalian cell, the growth media was supplemented with a radioactive amino acid. Now, which of the following molecules will be labeled? So, of course, since the growth media was supplemented by radioactive amino acids, the proteins being polymers of amino acids will be the first biomolecule which will be radiolabeled, right? The nucleic acids may also possess some degree of radioactivity, as there are certain amino acids that are used for de novo nucleic acid biosynthesis, but the major component which will be showing radioactivity would be proteins. So option number A is the correct answer. In the next question, the Indian student applied for postdoctorate fellowship in Singapore and he was asked to get tested for tuberculosis. Now he went to AIIMS, New Delhi for testing. The tuberculin skin test, which was the first test, turned out to be positive. However, the culture-based confirmation test revealed that he was negative of tuberculosis, which is the most likely reason for this observation. So the TB skin test that is performed by injecting a small amount of tuberculin under the top layer of the skin on your forearm if you have ever been exposed to TB bacteria. Your skin will react to the antigen and develop a fine red bump at the site within 2 days. Now the BCG vaccine, the Bacillus calmette goring vaccine, which is used for the prevention of TB, may cause problem in interpreting the tuberculin skin test, as the people who have previously been vaccinated by BCG may show false positive TB skin test. Hence, at times it is difficult to interpret that the positive TB skin test is either due to BCG or due to an actual infection of TB. So the TB culture test is preferred method for TB testing of people who have received a BCG vaccine. The TB skin test, this testing is not affected by prior, unlike TB skin test, this culture thing is not affected by prior BCG vaccination. So the correct answer for this particular question would be option number B, that the student was vaccinated by BCG and that is why it was testing positive from the tuberculin skin test, but it was for later on revealed that it was negative in the culture-based confirmation test. The next question is, why are MET and TRP often used to design DNA probes from amino acid sequences? Most of the hybridization studies that are used to locate a particular gene or other DNA or RNA sequence, they employ a tagged synthetic probe. These probes are often a small piece of DNA or RNA which is labeled with a radioisotope or a fluorescent bag. Now, the redundancy of the genetic code, that is the existence of 2 or more codons for a particular amino acid, presents a problem in designing an oligonucleotide probe based on amino acid sequences, right? The correct option would be they do not have a degenerate code. The methionine and TRP, they do not have a degenerate code, and that is why they are used in preparing the DNA probes from the amino acid sequence. The next question is, malaria is caused by? So the correct answer for this particular question is option number C, Plasmodium. There are 5 parasitic species that cause malaria in humans, and 2 of these species, Plasmodium falciparum and Plasmodium vivax, they pose the greatest threat. The next question is, the kind of covalent modification that occurs on both histone and DNA is? So both DNA methylation and histone modification is involved in establishing a pattern of gene repression during development. So the correct answer to this particular question is option number B, methylation, which happens both on DNA as well as on the histone modification. In the next question, a combination vaccine against 3 infectious diseases is? So the DPT vaccine, which is a vaccine for infectious diseases, diphtheria, whooping cough, and tetanus, is a combination vaccine amongst the given options. The vaccine components include diphtheria and tetanus toxoids that are either kill whole cells of the bacterium that cause the whooping cough. So the option number C, DPT vaccine, is the correct answer. The next question is asking that which of the following is the correct combination of marker enzymes which are used to identify different organelles during subcellular fractionation in eukaryotic cells. So the LDH or the lactate dehydrogenase enzyme is normally localized in the cytosol. and it is released by the damaged cell. Increased concentration of LDH in the cell supernatant is an indicator of the cell damage. The next enzyme, succinate dehydrogenase, is a mitochondrial marker which provides a variety of electrons that are needed for the respiratory chain process. Now, that particular process takes place inside the mitochondria. Let's talk about the acid phosphatase. Acid phosphatase is a lysosomal enzyme which removes phosphate from other molecules during digestion. And finally, the catalase is present in peroxisomes, where it decomposes harmful hydrogen peroxides either by converting it to water or by using it to oxidize other organic compounds. So the option A, which says cytosol LDH, Mitochondria-Succinate dehydrogenase, Lysosome-Acid phosphatase, and Peroxisome-Catalase would be the correct answer for this particular question. In the next question, a patient diagnosed with urticaria will have elevated levels of what? Hives or urticaria are red, itchy welts that result from a skin reaction. Acute urticaria is caused frequently by IgE-mediated allergic reaction, either to food or by an acute infection. So usually a viral respiratory tract infection or something like that induces urticaria. The correct answer for this particular question would be option number E, IgE. The next question is, Dolly sheep was created by the technique of somatic cell nuclear transfer, SCNT. This particular technique is used for cloning, in which the nucleus of the somatic cell is transferred to the cytoplasm of an enucleated cell. The SCNT was first used to create Dolly. So option number B, somatic cell nuclear transfer, is the correct answer for this particular question. The next question is, which of the following organism is exploited for transfer of gene in plants? The right answer to this particular question is option number A, Agrobacterium tumefaciens. This particular organism is a gram-negative bacteria which was established by H.J. Cohn that uses horizontal gene transfer to create tumors in plants. This bacterium is often regarded as the natural genetic engineer, and it became a very important tool in genetic engineering. The next question is, which of the following is an example of an attenuated vaccine? The examples of the currently available live attenuated vaccines against the viral infection include measles, mumps, rubella, smallpox, chickenpox, cowpox, yellow fever, influenza, and oral polio vaccine. So the correct option here is option number A, yellow fever. The next question is, which of the following method is not employed for affinity maturation of antibodies? Affinity maturation is basically a process where the immune system generates antibodies of higher affinity during a response to antigen. It is unique in being the only evolutionary mechanism known to operate on a molecule in the organism's own body. The in vitro affinity maturation has successfully been used to optimize antibodies, antibody fragments, on other peptide molecules, like antibody mimics. So the random mutation inside the CDR are introduced using radiation, chemical mutagen, site-directed or random mutagenesis. But the error-prone PCR, which is a universal method for introduction of such mutation, while high-fidelity PCR conceptually cannot be used for such a purpose. So option number C, the high-fidelity PCR, is the correct option for this question. The next question is, intrinsic fluorescence of GFP is contributed by. The intrinsic fluorescence of GFP, or the green fluorescence protein, this particular protein has 3 amino acids that form a fluorophore. It consists of residues 65 to 67, the serine, dihydrotyrosine, and glycine. These cyclized backbone of these residue, they form an imidazolidine ring. So the correct option for this particular question is option number A, presence of serine, tyrosine, and glycine. The next and final question is, which of the following sequences are not palindromic? Again, we'll have to write the particular sequence, and we have to write its complementary sequence and see if it is read same from the forward as well as the reverse direction. Once we've done that, we found that option number C doesn't read the same from both the directions. So option number C is not a palindromic, and the correct answer to this question would be option number C. Okay, thank you everybody. Please do watch this video. I hope it will be beneficial for you guys. And do not forget to like, share, and subscribe our channel.